Welcome to the DSNP klogW series. Today I'm just going to kick it off with some general comments about the series and about Zoom etiquette and so on before our speaker, Professor Roya Zandi, is introduced. So the first thing is just there's the division of statistical nonlinear physics, which I still find myself calling the group. So we actually became large enough recently, and you can see that in the Twitter handle. Has that changed? I'm not entirely sure. We should check. It has changed. So the Twitter handle has actually changed to DSNP and not GSNP. And we are a division now of the APS, and we focus on well, statistical and nonlinear physics, which is spelled out more on the APS website. I won't go into it, but you can get involved in all sorts of ways, including in the early stages as listed up there, and certainly by being a very committed participant in the seminar, for example. So here are our basic Zoom systems. At the moment, it is a webinar system. You are people other than the host and panelists. Oh, sorry, the speaker and panelists are sort of off screen, but you can put questions in the chat, and if they sound like or look like clarification questions, one of us will interrupt the speaker, and others we can save for the end. And maybe I'm not quite sure how it works now, but at the end certainly we'll have time for questions. But you may be able to ask the question directly as well. So we'll sort that all out when it comes to the end. But at the moment, at least, and please, this is the webinar format, so you probably won't be able to unmute yourself even if you wanted to. So leave it there for now. Okay. And now Shima will introduce.
Jack, did you want to make a quick pitch about the elections?
Oh, right. The elections are going on, and you should vote. is I guess the best way is what I mean. What I mean by the APS elections are going on. I don't mean national or international elections. So choose wisely.
Yes. So another way to get involved is to please cast your vote. So look for an email from directvote.net, and the elections are open until till next week, Monday, November 24th. And in addition to the offices that are open every year, that includes the chair line, members at large, and early career members at large. We, for the first time, have the opportunity to vote for a counselor, a divisional counselor, someone that's dedicated to our division. So yes, please do cast your votes.
Okay, so today we have Professor Roya Zandi from University of California, Riverside. She is a professor of physics and astronomy and the director of the biophysics graduate program at University of California. Dr. Zandi received her PhD from UCLA and then moved on to do a postdoctoral research at UCLA and then MIT. She became a faculty at UC Riverside in 2005. She's additionally an APS fellow through the Division of Bio, and her research is on computational self-assembly of the viruses. Without further ado, the floor is yours.
Okay. Thank you so much for the invitation and nice introduction. So you don't see my slides right now, do you? Okay, so let me share them. Now, do you see them? Okay. All right, thank you. So as Shima said, a large section of my research is about the viruses and especially how they form. So in this picture, you can see 3 different viruses. They are at different scales, and they are not at the same scale. But the first one, it's CCMV, is a plant virus. The second one is the herpes that you heard more about. It is a large virus, is much larger than CCMV. And this is a phage virus. It's a virus that infects bacteria. Even just with simple looking at them, you see something common among them. And you see that they have a very nice symmetric shape. And this was, this feature took my attention to understand why they assume such a nice symmetric structure. And it's not only viruses, many protein shells in nature, they have nice symmetric shells. So to just a brief introduction about viruses. They are composed of a nucleic acid genome, which can be RNA or DNA, and a protein shell called the capsid. So this is the blue line here is just mimicking the genome, and then this is the shell, the capsid shell. It shows inside the virus. The capsid protects the genome against the harsh environment between the cells or inside the cell. I don't know why for changing the slides. Okay. So I would like to tell you the life cycle of a virus. I chose Corona because most of us have been locked us at home for this virus. It's not, I am not going to talk about coronaviruses, but most viruses and the life cycle of most viruses are similar. At least for a physicist, there are some nuances between them. Okay, but this is a very general view of life cycle of a virus. So this is a coronavirus. It gets attracted to the cell receptor. So there are ligands on the virus that gets attracted to the receptor of this. This is the cell receptors. Then the cell engulfs the virus and the virus goes inside the cell. Open injury, and we don't know why and how it disassembles. There are some theories, and it might change from one virus to another. They think ionic strength changes or pH changes. But anyway, the virus releases its genome. The genome has the code for making the proteins that the virus needs. So the genome take over the cell machinery and then produces more genome and also produces the proteins that it needs to assemble. So in this case, the genome, coronaviruses have, the RNA of coronaviruses are 30,000 nucleotides. So it's a huge, I think it's one of the largest RNA viruses when it comes to its genome. And then, so this needs to be compactified, and it does compactified with nucleocapsid proteins that genome RNA is negatively charged. The nucleocapsid proteins are positively charged. They compactify the genome. And then the structural proteins, the proteins that contribute to the formation of the shell, they package the genome and then make the virus, assemble by making the virus. They assemble, in this case, actually is budding. The genome buds at the region well inside the cell called the ergic. It assembles, and at the end, it exits the cell. So I totally simplify this process. We don't know much about assembly of viruses because, first of all, they are very small. The size of, I forgot to tell you, this is 100 nanometers, and this is one of the largest viruses. The CCMV that I showed you was only 30 nanometers, so these are small, and they are found well inside the cell, meaning that it's very hard, the imaging of the intermediate, and the intermediate are transient. So our knowledge about formation of the viruses is very elementary because of the physical barriers like imaging and the time scale of the assembly. The viruses, I mean, there are many ways to classify the viruses, but what is amazing that they can be some of them that has been reconstituted in vitro. So this is a tobacco mosaic virus, TMV. This is the first virus that was reconstituted in vitro. So if you mix the capsid proteins of purified capsid proteins of TMV and its genome, and these experiments were done 70 years ago, fully infectious particles form. And as you see, they have rod-like shape. 12 years later, the first spherical virus was reconstituted in vitro. Once again, you mix purified protein with the genome, and then again, fully infectious particles form. So, and these processes, you know, it's formed spontaneously, no external forces, nothing. It's just a genome and purified proteins, and then you get in vitro fully infectious particles. So I would like to focus on the spherical viruses because as I showed in the first slide, the symmetry, it's what attracted my attention to viruses. So this is an icosahedron. It has 20 equilateral faces. These are the elements of icosahedral symmetry. So when I talk about icosahedral symmetry, these are the elements of icosahedral symmetry. It has 15 two-fold rotation axes going through the middle of each edge. It has 10 three-fold rotation axes going through middle of each triangle, and it has 6-fold rotation axis going through middle of each vertex. So for the rest of the talk, when I talk about icosahedral symmetry, I am talking about these three elements. These are the elements of icosahedral symmetry. So you can immediately see that in addition to icosahedron that we see here, like soccer ball, has a structure with icosahedral symmetry. These three elements, you can see that the soccer ball has also these three symmetry axes. So this is the CCMV that I showed you, the plant virus that I showed you in the first slide. So again, it looks very much like a soccer ball. It has 12 pentagon. It has 20 hexagon. And they all, it's made of 180 protein subunits. And all the proteins, all the monomers, all the protein that make the virus, they are made of the same protein. But they form a slightly different configuration because some of them has to sit on the 5-fold axis, some of them has to sit on the 6-fold axis. Actually, this is more 3-fold because red and green, they are not exactly sitting at the same positions. So if you look at it, there are 3 different positions at this virus. So that's where the triangulation number T = 3 comes into account. So the viruses are classified by triangulation number. And in this case, we have 3 different positions and is T = 3. Because I will talk about T numbers, I give you a little bit more information about triangulation number, T numbers. So here are different T numbers, triangulation numbers. Every single closed shell made of pentagon and hexagons must have 12 pentamers, and it has 10 times T - 1 hexamers. Pentamers because each protein subunit is called monomer, so 5 of them is pentamers. So I switch between saying pentamers and pentagons, but I mean the same geometry. So here is T = 1. It has 12 pentamers, and you can see if you put T = 1, you can see it's 0 hexamers. but it has icosahedral symmetry, the elements I told you about. And this is T equal 3. Again, if you put here, you can see it has 20 hexamers and 12 pentamers. T equal 4, it has 12 pentamers and 30 hexamers. And T equals 7, it has 12 pentamers and 60 hexamers. So all of these have only 12 pentamers, and but the number of hexamers changes. This is like quantized. In order to preserve the icosahedral symmetry, you should have a specific number of hexamers. For those of you who are familiar with Euler's theorem and the relationship between faces, edges, and vertices, you can get more pentamers, but then you need to have heptamers. But viruses, they don't form heptamers. They either make pentamers or hexamers. And T numbers is the index that classifies their size and number of hexamers. Another thing that I would like you to know about viruses is that they are flexible, because one might say they make icosahedral symmetry because it's built in the shape of their protein subunits. But this is not the case. So here it's an experiment done with one of my collaborators in 2007, Bogdan Dragnia. He mixed the capsid proteins of another plant virus, mosaic virus, capsid proteins, and functionalized gold nanoparticles. So, and then he changed the size of the gold nanoparticles. The natural size of BMV is T equal 3, but then he observed that as they change the size of the gold nanoparticles, the size of virus changes. And in this case, it goes from one T number to another T number. So it means that all of them, when they change the size of gold nanoparticles, they still have icosahedral symmetry, but the number of protein changes. But the symmetry is still there. More recently, he did another experiments, again with mosaic virus capsid proteins, but in this case, he used small nucleoacid oligomers, about 52 nucleotides. And you can see that capsid proteins, they don't make icosahedral symmetry anymore. So now he obtained this this structure, and you can see there is a row of hexamers and pentamers. And these structures, when two pentamers are sitting next to each other, so the capsid proteins are able to form structures that they don't have icosahedral symmetry. So it's not like built in the code of the proteins that when they sit, they have to, I mean, it's not engineered. They are not engineered to form icosahedral symmetry. So it's beyond the, so there must be another thing that they can form these shapes, but often inside the cell and in vitro, they form icosahedral symmetry. So how do we understand the assembly of symmetric shells? So first, I'm going to show you our computer simulations. And then I use continuum elasticity theory to explain the result of the computer simulations. So because the viruses are made of pentamers and hexamers, so we focus on trimers, on triangular subunits, because they can make both pentamers and hexamers. So because we said that elasticity is, important, we think that is important. So we assume that there is, so this is a trimer made of 3 protein subunits. So this is our basic building blocks. It has a bending energy. The preferred angle between 2 triangles is theta naught. So as the capsid grows, these triangles are added to each other. So the preferred angle is theta naught, but as it grows, the angle between them, we call them theta ij, between two adjacent, two neighboring trimers. Kb is the bending modulus. Then the equilibrium size of the, then there is a stretching energy involved, and the equilibrium size of each length is B naught, the size of the triangular subunit. And then there is a stretching modulus because we assume that it's elastic. So there is, this is like a spring constant Ks. So you can immediately see that there is an important dimensionless parameter comes out of this, which is the ratio of the stretching modulus to the bending modulus. And we like to make it dimensionless, so we multiply by the B naught, the size of the triangle, equilateral triangular square. And so this is almost like a Foppel-von Karman So immediately you see that if gamma is large, means Ks is large, so subunits resist deformation from the shape of an equilateral triangle. And when gamma is small, it means Kb is large and Ks is small, so subunits can easily be deformed. So an important step here is to make pentamers and hexamers. So this is the shell. These triangles are growing on a curved space. So if it were flat, that would be easy. So you add 5 triangles, then you will have so each of them, it's 60 degree here is equilateral triangle, and you can easily make a hexagonal sheet. But when it's growing on the on a curved space, so you can see here I have 5 triangles. Then there is an opening angle here. This is because of the curvature is not 60 degree anymore, this opening angle. Then we have an option. We can either insert a triangle and make a hexamer, or we can join these 2 edges and make a pentagon. So therefore here we have 2 options, making a pentagon and hexagon, and we do this by calculation of the energy. We see if the elastic energy, if the elastic energy goes, which of these configurations does the elastic energy prefer? So this is first we start with a very crude model just to see what we get. So, and we have only elastic energy. So we add protein subunit 1 by 1, and this is growth along the minimum free energy pathway. So it's irreversible. We just look for the position. So we add, we assume that this is a slow process, add proteins 1 by 1, and then let the system relax. And we assume that the attractive interaction is weak. So the protein has the option to find where to be added. So, but where the place it chooses is the place in which the subunit maximizes the number of intermolecular bonds. So we assume that there is some interaction. So they are going to sit where the number of intermolecular bonds are maximum. So in this case, all the edges are equivalent. But in the next one, so randomly we add a triangle. But after that, so then we can see that these 2 positions are almost equivalent when you add one. So we add another one here and then here. And then after that, we are going to, I mean, the subunits will be added to fill out this row. And again, we continue this way. And once it's added, once protein added, we won't remove it. When we are adding it, we find where it maximizes the number of intermolecular bond. But after addition, when it added, it's irreversible, it's finished. So as I said, it's a very crude model. It's along the minimum, the free energy path, but it's irreversible. Obviously, we didn't expect to get icosahedral symmetry in this model. So you can see here, for example, we added 5 triangles. And then here, the opening angle alpha, as it grows on the curved space here, this one became a hexamer. It keeps growing. At some point, alpha starts getting smaller and smaller. And here you see we have 1, 2, 3, 4. And then we make a pentagon here. It doesn't become a hexamer. But you can see the position of pentamers is such that the symmetry is broken and we don't have icosahedral symmetry, as we didn't expect in such a crude model to get it. But what surprised us, we decreased the radius of curvature, the same model, irreversible, along the minimum and free energy pathway. We added triangle as in this movie, 1 by 1, and then you see, we don't remove them. Once added, it's done. The red ones are pentamers and the blue one are hexamers. And we can see that as we add them, the pentamers form exactly as the shell is growing. The pentamers form at the position where the shell is completed. They are sitting at the vertices of an icosahedron. So that was quite puzzling for us, knowing that this is such a crude model. Then we made just a phase diagram as a function of focal one Karman number, which, as I told you, is the ratio of the stretching to bending moduli, and the preferred curvature. I told you that protein, there is a dihedral angle, which is related to the radius of curvature. So this is a 2-dimensional phase diagram as a function of gamma, focal one Karman number, and the preferred curvature. Each solid color corresponds to a different structure. So I have the structures here, and you can see A is the T=1, the smallest possible icosahedral structure, and G is T=3, and the plant viruses I showed you, like the first slide, CCMV, it's a plant virus, and it has T=3. And here it's another icosahedral virus, which corresponds to hepatitis B virus. And this one, F, is actually the structure that my collaborator, I told you, using small nucleotide. I showed you that capsid protein can form structures that they don't have icosahedral symmetry, but still is a closed shell, and is that structure. I have another talk about this structure. I find it very interesting, the way the symmetry and stress is distributed, but it's subject of another talk. Here we focus on icosahedral viruses. One thing that I would like to tell you is that, so we saw that this one was irregular. So we saw that the small, when the shell, maybe I should say here. So when the radius of the spontaneous curvature, or the radius is small, we saw icosahedral symmetry. But when we increase this radius, as in this 1D simulations, we get irregular shapes. So as the curvature increases, we get irregular shapes. And we can see actually at this position, at this curvature, we get a conical shape, which looks like the capsid of HIV, that, again, we don't talk about it much today. And even when increased radius of curvature, you don't get even disclination pentamers. You just get a cylindrical shape. But these are not the structure that I'm going to talk about, but I just wanted to tell you that as the shell gets larger and larger, we won't get a symmetrical structure. We don't get symmetrical structures. But this is not what we see in nature. In nature, so in the phase diagram, I showed you the maximum T number. And again, remember, T shows you the number of hexamers. T minus 1 and times 10 shows you the number of hexamers. And so here you see thousands of proteins, they join each other and form icosahedral structures. And so all these structures, like herpes, is T equals 16. And so you have to, in order to know how many proteins you have, you need to multiply by 60 because to get the number of proteins in each protein shell. And they all come together, and the distance between these red pentamers that's shown, no matter how big they are, is equal. You see the distance is equal, so they have icosahedral symmetry. If you carefully look at these structures, you can see that they have, all of them, they have something in addition to the shell and the genome. They have either scaffolding protein or they have inner layer. So it seems that there is something that is helping their curvature. Many of you might have seen this slide, this movie online. So these are it's 12 pentagons. And this is, if you shake it, it's like adding thermal energy to these magnets that they have been cut to make an icosahedron. So if you and I, we sit down and make an icosahedron with 20 pieces of equilateral triangle, we can put them together and add them one by one. It takes a time, but we can do it too, like this, shaking this these 12 magnets. But if we want to make a large shell with 500 pieces, it's going to take us a lot of time to make an icosahedral shell. So then we said, what should we do? I mean, what does nature do to get these structures? So my student thought about the yoga ball that we had at the office. So we started to see how the curvature of the core helps us to make icosahedral symmetry. So then they started making, just put them together around a yoga ball, and it only actually took an hour or 2 hours, and just putting all of them together, and just icosahedral symmetry came out of it. So the curvature of the ball, of the yoga ball, commensurated with this T structure, and we could easily get it. So we thought that, okay, maybe we can do similar thing. We added a core to our, because we couldn't get more than T equal 4 in our simulation. Again, T equal 4 has only 30 hexamers. And then this is T equal 13, it has 260 subunits. So we said, let's add an inner core. And the only thing is that the core interact with subunits through what is Lorentz-Jones particle, which is just attractive, just add a small attractive interaction between subunits and the core. And then again, we did irreversible, meaning that once a pentamer formed, that's it. We cannot move it, it's done. And again, to our surprise, every single pentamer formed at the vertex of an icosahedron. Even though this is a growing shell, it has a boundary, moving boundary condition. We found again icosahedral symmetry. when we look at the stretching energy and the bending energy of these structures, so we saw that the bending energy, I mean, it's over around the sphere, so it's monotonically increased as expected. But what we saw that was interesting, we saw that stretching energy drops each time a pentagon forms. Each time we form a pentamer, we saw that the stretching energy that I showed you, that was very simple, it drops. So to understand this one, we resorted to continuum elasticity theory. This part is the elastic energy, and this is the bending energy. And this part does not do much for us now because it's growing. We assume that the shell is growing over the surface of a sphere. So this part doesn't contribute. But then we focus on this one. Mu and lambda are Lamé coefficient, and U is a stress tensor. Minimizing the elastic free energy actually is done by Sung and Nelson in 1988. We get this bi-Laplacian equation where chi is the area stress function, S is the disclination, is in our model is like pentamers. And each disclination, because we have pentamers, for those of you who are familiar with this equation, disclination has the charge of pi over 3. K is the Gaussian curvature. Again, in our case is easy because it's a sphere, so it's just 1 over r square. And then, so then once we have chi, we can put it back, and then we calculate the free energy. And y is the Young modulus, which is as a function of, we can write it as a function of Lamé coefficient. I'm not going through the details of math. The appendix of this paper has all the details. But I just want to tell you the difficulty of these equations, and I am so proud of the former student, C.U. Lee, that solved this equation. So this is a Laplacian in spherical coordinate, and then so we solve this by Laplacian. In the presence, this is like a delta function of the disclination with the stress-free boundary condition. And we have been able to solve this equation actually analytically. I mean, the solution is a few pages of Mathematica, but it has a closed form. And we find that it involves four parts. One is just due to curvature, the Gaussian curvature, and then is the defect and the Gaussian curvature interaction. Then each defect by itself has some self-energy. And what is the most important contribution of this work is that the interaction between two pentamers or two disclinations. So this interaction is, it is defined, I mean, obviously is screened by the Gaussian curvature, defines the position of pentamers. So this row is the solution. It's our irreversible simulation, the first row that I showed you. Blue are hexamer, yellow are pentamers, and reds show the edge of the shell, and it's growing on the core. And the bottom row is the solution of our elastic equations. So we follow the same path as the simulations. We grow the shell, and then we find where we add the disclination, where the energy drops. So these are the energy contour plots for the newest disclination. So the larger the dots are pentamers, and the larger white balls are the newly added pentamers or discalinations. So we looked to see where to add the pentamers, where the energy drops. And we see that an amazing match between the solution to the elasticity equation and our simulation. And this was also the same way. It was once we formed these discalinations, we froze their position, and we grew the area similar to our simulations. And we saw that the discalinations appear exactly where it was appeared in our simulation. So it seems that there is a deep energy well. This is a growing shell. We know if we have 12 electrons and we put them on a conductor, a spherical conductor, they're going to sit on the vertices of an icosahedron. But here, this is a growing shell, and it has a moving boundary condition. So this is amazing that these discalinations already appear at the positions that we won't move them later. But then when the shell is complete, they have appeared where, when the shell is complete, we have an icosahedron. So this is, we did another work where we actually minimized. So that was, again, irreversible. But in this movie, at each step, we could move the discalinations, and we saw that obviously icosahedral symmetry has the lowest minimum energy structure for a growing shell. So I am going through this slide fast because I just want to show you that we did, so the simulation that I showed you up to now, they were all about the irreversible simulation. But then we decided, now that we understood the role of the elasticity, we decided to add attractive interaction between the proteins. So we have a hydrophobic interaction in addition to the elastic energy that I showed you before. And then there is attractive interaction between the triangular subunits and the core. And so as in the irreversible simulation I showed you, but then we did Monte Carlo simulation in grand canonical ensemble, when we can add subunits or we can remove subunits. So there are 3 Monte Carlo moves. We can, add one subunit and remove. The subunits can diffuse around the growing shell, or we can merge or disjoint. A pentamer can be attached or again detach, and so this is the 3rd move. So we had 3 Monte Carlo move. So this is completely reversible. And we wanted to see, now that it's reversible, we wanted to see the role of the elastic energy. So here you see that, for example, here in this case, I start with this one, we have 2 pentamers. A shell has been growing. We had too many subunits, or we built it just artificially, where 2 pentagons, 2 yellow ones, are sitting next to each other. This axis shows us the elastic energy, and this is like the time in Monte Carlo simulation, the frame number of steps in the Monte Carlo simulations. So you can see that when 2 pentamers are sitting next to each other, the elastic energy, A, is very high. But the hydrophobic interaction, this axis is hydrophobic interaction, again, versus the number of steps. You can see that the hydrophobic is very low because everything is attached here. So there is an attractive interaction between the triangular subunits. So when, as we see that the shell actually repairs itself by that, even though this pentamer is well inside the shell and it's is far from the edge, many subunits dissociate, and then the elastic energy drops. Obviously, hydrophobic increases because they are not attached anymore. And then it becomes, in part C, you can see it repairs this node. It's going to become a hexagon at C. So then the hydrophobic drops. It doesn't drop to where it was, but then we have elastic energy that just goes slightly higher. So the elastic energy drops. So the shell doesn't like two pentagons to sit next to each other. So it can completely repair itself. And we showed that when two pentamers, are based on our elasticity theory equation, when two pentamers are next to each other, the energy just shoots up. And then that's why icosahedra, it just comes because as the shell grows, the distance between pentamer is such that when they form, they are sitting at the vertices of an icosahedron, the energy significantly drops. Okay, up to now, I was either talking about a core that helps with the curvature or empty shell with preferred angle. But in reality, there is a genome that is in vitro, sometimes completely flexible. And at the end, we have a completely disordered system, and we get a shell with icosahedral symmetry. And that was a puzzle for many years for us, that how could we go, even knowing that icosahedral symmetry is the lowest energy structure, how could from such a mess we can go from a disorder to an ordered structure, and inside the cell, in vivo and vitro, under many different ionic strength, different conditions, ionic strength, pH, we can still get this structure. So we did MD, for this one, we did MD simulations. Again, we focused on triangular subunits. For our most MD simulation, they have done with rigid subunits. So we also used triangular subunits that they are rigid. But we assume that, and they attract each other, these rigid subunits. There is attractive interaction between them on the edge. But what is the novelty of this simulation is that we assume that when they join each other with certain probability, it's like they go through conformational changes, and then an elastic bond form between them. So this conformational switching mimics an allosteric response where binding activates new interaction modes. So that's the role of this elastic. So because in nature also, the proteins, when they interact with each other, they go through conformational changes, they expose maybe more hydrophobic part, and therefore, based on this thinking, we assume that when they join each other, then the elastic triangles that we had before, we added around them. So these are the rigid one, and then now they are interacting with certain probability. They have also elastic property, because we learned that we need elasticity if we don't engineer it 100% to form icosahedral symmetry, and they are flexible, that they can form other structures too, then we need definitely icosahedral, to get icosahedral symmetry, we need elasticity theory, elasticity property. So here is, for example, an step in our MD simulation. So here you can see we have 3 triangles that 3 subunits that they have already became show elastic properties. And as we said, with certain probability, they can go back and forth between becoming rigid, and still they attract each other, but this has transitioned from an elastic subunit to a rigid subunit, and vice versa. So during, as I show you the movie of the growth, you will see that they can switch between elastic and rigid. And when they become rigid, it can stay here, back to elastic, or just move away, diffuse away back to the solution. And then there is attractive interaction. This is only, I showed you 4 monomers of the genome. There is attractive interaction between the subunits, either rigid or elastic, it doesn't matter. There is attractive interaction between the subunits and the genome. So here is the movie. So this is the genome. This is a linear genome. And these are the subunits that I told you they can, when they are diffusing, for diffusion elasticity theory, it's not important. I mean, the fact that they are elastic or not is not important. And then you can see it's such a mess. When you see the green triangles, you see that they have already switched to the elastic triangles, and they go back and forth. And I would have never thought that such a mess that you can see, it's completely messy. And it takes a while to fix all the wrong positions that triangles or pentamers formed in the wrong positions. It takes a while to overcome all the energy barriers and the mess that we had, because the proteins were attracted everywhere. And then at the end, because of elastic property, we have been able to get the right symmetry. So these are snapshots of simulations of all these. They have the same parameters. So this shows the fluctuation. So this is, you can see that it takes much longer in this case for the shell to starting at all this starts from you know, this is after 800 steps or 800 time units. You see, they all have very disordered structures, but as time passes, they start, I mean, they all form at the end of the day icosahedral symmetry, but the pathway is different for the same parameters, but as expected, the end results are the same. There is obviously energy barriers. Here is the energy versus time. You can see that there is an energy barrier because once it's formed, it's hard to dissociate and reassociate and fix. Fixing the shell sometimes, I mean, in this one, you can see fixing the shell takes more time if they are formed in the wrong position than actually just something forming a closed shell, but with the wrong symmetry. But no matter what, if we wait, at the end of the day, we get the right symmetry. And this plot shows you the RG radius of gyration of the both proteins. This is for the genome, the orange line, the radius of gyration of the genome. You can see very quickly drops to its final value, and this is the number of trimers. And we can see that it go for a t equal 3, we need 60 triangles. So it's 60 triangles. It's more because some of the triangles were assembled inside the capsid. That's why we have a higher number.
Zoya, I'm sorry to interrupt, but there's just a note that there's 5 minutes left in the official time for this.
Okay, thank you for the reminder. I'm almost done. So this is the phase diagram. This is the number of protein subunits, and this is the length. And then, as you can see, as the length, depending on the length, we can get different structures. And then and many of these structures like doublet and incomplete shells and different T numbers have been also observed in the simulation, in the experiments. So this is the summary of I told you. I showed you that elastic energy is important, and as a shell grows, discontinuation appear on the vertices of an icosahedron, even as the shell is growing. I showed you that even with MD simulation, when we add elastic property, we have been able to get the icosahedral symmetry that packages the genome, a shell that packages the genome, and the elastic energy can repair. If we allow the reversibility, the shell can repair itself, and at the end you get the right symmetry. And I would, at the end, I'd like to thank all the group members who contributed to the to this work over the years. I showed you over decades of work. Especially, I want to thank CU that all the latest simulations were done by her, and now she's an assistant professor herself. And I would like to thank all of you who came to this talk. Thank you.
Thank you.
Should we invite people to put questions in the Q&A? There is one question. Thank you, Roya, for a beautiful talk. Can we jump into questions?
Sure.
There's a question in the Q&A. This is John Beckhofer. John, shall I just read the question, or would you like to jump in and?
Do you want me to
Your talk is, John Beckhofer asks, is your talk focused on static structures and how they form? Is it interesting to look at the eigenvalues, eigenvectors of the deformation modes? Is much known about the dynamics of such a shell?
No, we haven't. This is another step that hopefully we would like to do. No, we haven't looked at the eigenvalue. We haven't looked at the dynamics equations. So everything, even though when we are growing the shell, in the elasticity theory, we froze it, and then we calculate the minimized energy, and then we grew the shell, and again. So this is another nice extension of this work to add dynamics to the equations. Please
feel free to put things in the chat. Actually, if I could ask, I felt a little bit confused about what in the case of the yoga ball and the nanoparticle, it's clear you provide R0 as one of the parameters to the shell, or at least it seemed clear to me. But I was very confused by the end simulation, because even the fragments of shell that are incomplete and are grabbing only part of the genome, they seem to have the right radius of curvature.
You mean the MD simulations?
Mm-hmm.
So there is a dihedral angle between triangles. When they join each other, they join with the preferred angle. And then that preferred angle defines not 100%, but there is a flexibility around it because there is a bending modulus. But that defines the size of the final shell.
So
preferred angle.
So both R0 and gamma in your model are sort of fixed by the shell pieces themselves.
Right. But you saw it's almost like a harmonic potential. So, and it has some
Plus
flexibility.
fluctuations, yeah.
Right. And if you look at, I mean, the phase diagram, it's, let's see. If you look at the phase diagram that I made for this, you see for a large number of radius changes here and here, from here to here, you get t equal 1, and from here to here, you get t equal 3, actually from here to here, depending on gamma. So for a large range, you get icosahedral symmetry. And that's what MD, even though there is some flexibility, because icosahedral symmetry is so preferred. So that's why at the end, they pick that structure.
If there's questions, as Shima put in the chat, please, yeah, jump in, Doug.
Hey, Roya. So your talk focused on the self-assembly as would happen in vitro. And I wonder how that compares and contrasts with self-assembly in vivo. I mean, is it really the same process, or maybe is it possible that self-assembly is occurring while the cell is producing all of the ingredients? How does that compare and contrast?
That's actually one of the question of virology, that how the capsid proteins inside the crowded environment of the cell packages their own genome. And there are many, many theories about suggestions. But what I showed actually, it's not really, I focused on, I showed you the in vitro experiments, but even in vivo, even if you say that they all form in the proteins and the genome, and all the proteins are formed in the vicinity of each other, still the protein have to join, it has to package inside the cell. So it's the same physical rules has to be there. Otherwise, how it would assemble, package the genome, make a closed shell, and the shell has the right symmetry. So regardless of where in the cell this process is happening, one question that they asked, they said, because inside the cell there are a lot of other RNAs, why it packages its own RNA. One, there are several ideas. One, they said what you said, they happen in the proximity of each other, so that is close. That's why it packages its genome. Another one is because the size of the genome is the right size for the curvature of the protein. Another idea is that there is something called like packaging signal. There are certain secondary structure or tertiary structure of RNA that it's like key and lock with the protein subunits, and that's why it picks the right genome. But that's for picking the right genome. It's not about how the shell assemble and how from disorder goes to the order and packages. It's just about picking the right genome in the crowded environment of a cell.
Yeah, one could imagine that the self-assembly of the interior and the self-assembly of the shell are coupled.
But how is
or
this?
happen sequentially, right?
But our simulation is also, if you don't put all the proteins right at the beginning and add it slowly, you get the same results. Maybe I am not following why this is different in vitro, in vivo. It's the same proteins are joining each other, no matter and then they package the genome. Either it's in vivo or in vitro or anywhere in the cell.
There's a question from the audience. Please, yeah, go ahead.
Oh, sorry. I have a follow-up question. So I wonder, so I do see that there are some like organizing on the structure once the structure is formed. I just wonder, like, in terms of the genome packaging, is it possible that it happens after the capsid is formed? Like, and I wonder if that can be like studied via simulations, like putting some like chemical fuels and to see how the genome is inserting into this capsid.
Yeah, very good question. Not in case of RNA viruses. This happens in case of DNA viruses. So DNA, because RNA is flexible, and this is a co-assembly, as I showed in the MD simulations. But for DNA viruses, DNA is pretty stiff, and the size of, the persistent length of DNA is 50 nanometers, and the size of these viruses is small. There have been a lot of simulation and experiment to show that for packaging of DNA, you need a motor protein, meaning that a motor has to push the DNA inside the capsid. Is not the co-assembly. So the capsid forms, and then there is an opening, and then the motor proteins pushes the DNA inside the capsid and then forms. But in RNA viruses, that's why actually I mostly have been studying RNA viruses. It's the co-assembly, it's the minimum free energy structure, because you put them in solution and in vitro, they assemble spontaneously inside the cell without any ATP, any external forces, any help. They assemble. And that's why in simulation, we have been able, without any using other things like motor protein or other external help, they assemble. We have been able to do MD simulation that packages the genome.
Mm-hmm. Thank you so much. I also wonder, like, how the entropy is changed, or what's the role of entropy play in this spontaneous like process.
As you saw, we go through an energy barrier. So entropically, we go through an energy barrier because, yeah, once they form, it's very hard to dissociate and repair, the shell repair itself. But yeah, but it seems that the elastic energy of the icosahedral shell overcome the entropic barriers and dissociate and reassociate again. So the elastic energy wins at the end of the day, at least within this range.
Thank you.
Parameter. You're welcome.
We have another question. I'm going to unmute you. Go ahead.
Hi, thank you for your talk. To piggyback off of that last question, I was wondering if the chain inside of the finally formed icosahedron is maximizing the number of contacts with these triangular tiles, or if it's sort of packaged in a ordered arrangement once the icosahedron does form?
Yeah, that's a very good question. Because right now we are looking at the disassembly of these shells. And the most important thing actually is the number of contacts for disassembly to understand where it starts. But the thing is that we did not really study it. We are doing it for disassembly. For assembly, we did not study to see that the final structure, what is the distribution of genome inside the capsid for the final structure, but based on the images of viruses, not on our simulation. the genome sits next to the shell, next to the and close to the capsid proteins because of electrostatic interaction between the capsid proteins. There are positive charges on them and negative charges on RNA. So that's why the number of contact is maximum if as many contacts as possible because of electrostatic interaction between genome and positive capsid proteins.
Okay, interesting. And quickly to ask, you mentioned disassembly. How are you triggering that in the simulation? Once you get to the shell, do you turn off the potential energy functions or something?
Something similar like that. They have done it in vitro with, and they think that is similar mechanism is happening in the cell by changing pH, and pH changes the number of charges on proteins. So either by changing the number of charges on the proteins, which weaken the electrostatic interaction, or also, again, hydrophobic, the effective protein-protein interaction, because when you change pH, you change the number of charges on protein, so you change the effective protein-protein interaction. So that's how we trigger in the simulations.
Okay, thank you so much.
You're welcome.
We will take a last question from Shintang Jiang. Will you?
Hi, Professor. Thank you for your talk. So, as you mentioned in your work from the growing of the shell on a spherical core, so you mentioned that that process is irreversible, but in reality, like or at least in the MD simulations, the assembly process, like if two capsomers are attached to each other, like there are chances that they would, like, or I guess what I mean is there chances that like they are going to even the bomb forms, is there chances that the bomb will be break and then the attached capsomers will be like disattached?
Yes, thank you for your question. I guess I wasn't clear enough. So the simulation that I showed at the beginning, I wanted to show you that even with a crude model, when there is no dissociation, like the one actually I showed, I have on this slide, the next slide, I think, yeah, I in this one, I showed you that even in this one, no bond is broken and I get icosahedral symmetry. But for the MD simulation that I showed at the end, I removed, I mean, I don't have this rule anymore. They can join each other and then they can dissociate because there is an attractive interaction, there is a temperature, it's an MD simulation. So, and as I think we can, you can see here that as it's growing, these are associating, and these, I mean, this, for this mess to become a shell, so a lot of proteins, they have to dissociate and associate and reassociate and move. So yeah, in this MD simulation, they associate and dissociate. But what I wanted to show that the energy of icosahedral, it's so dominant that even with a much simpler model, we have been able to get, which it was irreversible, we have been able to obtain icosahedral symmetry.
Okay, thank you. That answers my question. Thank you very much.
Maybe we take the one last question that's already in the Q&A. And at that point, maybe we can thank Roya again, since we're at 1:15. The question is, does the chirality of the building block play any role in the final structure? Can it break icosahedral symmetry?
Yeah, our protein, yeah, we don't take into account chirality of the proteins. But yeah, but for in this case, the protein, even if they are they have chirality, what is important is that when they join each other, it's the hydrophobic interaction between them. So if the chirality changes, the hydrophobic interaction weaken or make it stronger, that might help, that might change the final structure. Otherwise, I know that like T = 7 is these T = 3 is not the chirality, it doesn't have even chirality, but T = 7, it has chirality, but the proteins, the chirality of protein hasn't had that impact on the final structure. But this simulation cannot answer that question because we don't have it in our model.
Thanks, Roya. Maybe this is a good time to all thank
you.
Roya again for a wonderful
Thank you.
talk.
Thank you.
And thank you all for attending.